Restriction enzymes. Key molecular tools in genetic engineering. Introduction. Restriction enzymes, also known as restriction endonucleases, are proteins that play a pivotal role in molecular biology and genetic engineering. These enzymes act as molecular scissors, cutting DNA at specific sequences known as recognition sites. Discovered in the 1960s, restriction enzymes have revolutionized biological research, enabling scientists to manipulate DNA with precision and paving the way for advancements in recombinant DNA technology, cloning, and genome editing. This comprehensive exploration of restriction enzymes delves into their discovery, types, mechanisms, applications, and impact on science and medicine. Discovery of restriction enzymes. The discovery of restriction enzymes was a milestone in the field of molecular biology. In the early 1950s, researchers observed that certain bacteria could resist infection by bacteriophages, viruses that infect bacteria. Werner Arber, along with colleagues Hamilton Smith and Daniel Nathans, identified that this resistance was due to enzymes capable of cutting foreign DNA. These findings earned them the Nobel Prize in Physiology or Medicine in 1978. Arber proposed that bacteria possess two systems: a modification system that protects their own DNA through methylation and a restriction system that degrades invading DNA. The restriction enzymes isolated from bacteria became the foundation for modern molecular cloning techniques. Structure and function of restriction enzymes. Structure. Restriction enzymes are typically composed of one or more polypeptide chains. Their active sites are structured to interact specifically with DNA molecules, recognizing particular sequences and catalyzing the cleavage of phosphodiester bonds. Recognition sequences. These enzymes recognize short, specific DNA sequences, usually 4 to 8 base pairs long, called recognition sites. The sequences are often palindromic, meaning they read the same forwards and backwards on complementary strands. For example, EcoRI, a commonly used restriction enzyme, recognizes the sequence 5' GAATTC 3' 3' CTTLAG 5' mechanism of action. Once the enzyme identifies its recognition site, it binds to the DNA and induces a conformational change. This positions the enzyme to cleave the phosphodiester bonds within or near the recognition sequence. Depending on the enzyme, the cut may produce blunt ends or sticky ends, overhanging single-stranded DNA regions, e.g., EcoRI. Blunt ends. Straight cuts with no overhangs, e.g., HAII. Types of restriction enzymes. Restriction enzymes are classified into several types based on their structure, recognition sites, and cleavage patterns. Type 1 recognize specific sequences but cleave DNA at random sites far from the recognition sequence. Require ATP, S-adenosylmethionine, and magnesium ions. Less commonly used in genetic engineering due to their unpredictable cleavage. Type 2 recognize specific sequences and cleave directly within or near the recognition sites. Do not require ATP. Most widely used in molecular biology. Type 3 recognize specific sequences and cleave a short distance away from the site. Require ATP and magnesium ions. Type 4 target methylated or modified DNA. Play roles in host defense systems and are increasingly studied for epigenetic applications. Type 5 utilize RNA-guided mechanisms for cleavage, e.g., SAS enzymes used in CRISPR technology. Applications of restriction enzymes. 1. Recombinant DNA technology. Restriction enzymes are fundamental in creating recombinant DNA molecules. By cutting donor DNA and plasmid vectors with the same enzyme, scientists generate compatible sticky ends, allowing the insertion of foreign DNA into vectors for cloning. 2. Gene cloning. In gene cloning, restriction enzymes facilitate the insertion of target genes into plasmids. These recombinant plasmids are then introduced into host cells, enabling the production of proteins such as insulin or growth hormones. 3. Molecular diagnostics. Restriction fragment length polymorphism, RFLP, analysis employs restriction enzymes to identify genetic variations and mutations. This technique has applications in disease diagnosis, forensic science, and ancestry studies. 4. Genome mapping. Restriction enzymes assist in creating detailed maps of genomes by cutting DNA into fragments of known sizes, which can be analyzed using electrophoresis. 5. CRISPR technology. While CRISPR is primarily guided by RNA, Many SAW proteins function as restriction enzymes, cutting DNA at precise locations. This technology has revolutionized genome editing, enabling the correction of genetic defects and the development of gene therapies. 6. Epigenetics type 4 restriction enzymes, which target methylated DNA, are valuable in studying epigenetic modifications such as DNA methylation patterns associated with cancer and other diseases. Impact on science and medicine. Advancements in biotechnology. The ability to cut and splice DNA has led to significant progress in biotechnology. Restriction enzymes have enabled the development of genetically modified organisms, GMOs, industrial enzymes, and biopharmaceuticals. Gene therapy. Restriction enzymes are integral to creating vectors for gene therapy, offering potential cures for genetic disorders such as cystic fibrosis and sickle cell anemia. Personalized medicine. Restriction enzymes contribute to personalized medicine by enabling precise genetic modifications and diagnostic techniques tailored to individual patients. Forensic science. In forensic investigations, restriction enzymes help generate DNA fingerprints, which are unique to individuals and used for crime scene analysis and paternity testing. Limitations and challenges. Despite their widespread utility, restriction enzymes have some limitations. Sequence specificity. They only recognize certain sequences, limiting their application for non-standard sites. Off-target effects. Occasionally, restriction enzymes may cut unintended sites, complicating experiments. Stability. Enzyme activity may diminish under suboptimal conditions such as extreme pH or temperature. Cost. High-quality restriction enzymes can be expensive for large-scale applications. Future directions. Engineered restriction enzymes. Advances in protein engineering have led to the development of modified restriction enzymes with altered specificity, expanding their applications in research and medicine. Integration with CRISPR. Combining restriction enzymes with CRISPR technology could enhance precision and efficiency in genome editing. Synthetic biology. In synthetic biology, restriction enzymes are essential for constructing synthetic genetic circuits and novel biological systems. Environmental applications. Engineered restriction enzymes could be employed in environmental biotechnology for bioremediation and monitoring genetic pollution. Conclusion. Restriction enzymes have profoundly transformed molecular biology, enabling scientists to explore and manipulate DNA with unparalleled precision. Their discovery not only deepened our understanding of genetic systems but also catalyzed innovations in biotechnology, medicine, and forensic science. As research progresses, the potential of these remarkable molecular tools continues to expand, promising even greater contributions to science and society. Thank you for watching. Subscribe.